anatomists and physiologists. This video is another video in our immune system series. This is part 2 about adaptive defenses. So just to recap where we left off last time, we talked about innate defenses. Those are those things that we're born with. They're the surface barriers. They're the internal defenses that are pretty non-specific. Your textbook describes the surface barriers and the internal defenses as the first and second line defenses, and the adaptive defenses collectively are the third line of defense. The innate system is great because it's always good to go, but the adaptive defenses require priming or being introduced to the thing first. And there's two branches of the adaptive system. There's the humoral side and the cellular side. The humoral immunity, that refers to the body's humors, which is an old-timey word for body fluids. Body fluids. Things like the blood, the lymph. And it deals with B cells. So it deals with B cells, and antibodies are made by B cells, and then the antibodies swim around in the blood and the lymph where they bind to antigens to mark them for destruction. And that's done usually by things like phagocytes, the complement system, etc. Cellular immunity is named that way because it's cells, the lymphocytes, specifically T cells mostly, that are doing the immune thing. This branch targets cells infected with pathogens like virus, bacteria, parasites, cancer cells, that kind of thing. But this happens more directly by targeting the cell, or more indirectly by releasing inflammatory chemicals, things that activate macrophages, that kind of thing. Even though the innate defenses and the adaptive defenses are like different branches of the immune system, notice that a lot of this is happening simultaneously, and there's different major players of the immune system that act in more than one branch. But the major things that set adaptive immunity apart is the use of B cells and T cells. So B cells and T cells. But it's also specific for defined pathogens. We've seen those pathogens before, we know them. Additionally, the innate defenses, that leads to like this local inflammation, but with adaptive defenses, this is more of a systemic thing. It's not just locally defined to the infected site, but it's this more widespread approach. And then lastly, the adaptive defenses creates memory. That's the big one. Creates memory so that stronger attacks can happen with subsequent exposures. All of these things are really important so that we develop immunity. That's the adaptive part. Before we can discuss the specifics about the adaptive immune system, first we have to know exactly what antigens are. Antigens are antibody generating. So they're antibody generating substances. They're usually foreign invaders because they're things that the body considers non-self. There's some characteristics about antigens that I want you to know about. Antigens, they can be immunogenic. To be immunogenic, that means that when that antigen is around, they'll stimulate lymphocytes. So they'll stimulate lymphocytes. To proliferate, to make more. To proliferate. Another word for proliferate is divide. But antigens can also be reactive. Being reactive means that when the antigen is around, they'll react to the activated lymphocytes and the antibodies that are hanging out. So it'll react. So to react, will react to activated lymphocytes and or antibodies. Most antigens have both properties. So if you're both immunogenic and reactive, we would call that antigen complete, meaning that it cannot cause all this not so great stuff. And this is for most antigens. We would call them complete. Things like proteins, large sugars, lipids, nucleic acids, bacteria, fungi, viruses, pollen, all that kind of stuff. But some antigens are not immunogenic. If they're not immunogenic, then they would just be reactive. And we would call those incomplete. So if you're just reactive, say reactive only, or reactive but not immunogenic, that would mean we would call that antigen incomplete. Incomplete. What happens is peptides, nucleotides, hormones, smaller molecules can enter the body and link up with the body's own proteins. And this can cause an immune reaction that way, but it's not the smaller molecule causing it in totality. And that makes a problem because now the body is attacking its own proteins because that little thing bound to our own protein. So that would be an incomplete antigen. That little thing causing the issue would be called a hapten, so incomplete, or we'd call it a hapten. It has reactivity, but it's not immunogenic on its own. And some examples are things like poison ivy, animal dander, detergents, makeup, these kinds of things. Antigens also have specific parts that antibodies can bind to. These are called antigenic determinants. So they're parts antibodies can bind to. Let me show you a picture. Okay, so here we have this antigen, and on the surface of this antigen, we have those antigenic determinants. We have this one that looks like a house, this one that's kind of like an ovalish thing, and then this one that looks like a puzzle piece. So those are the antigenic determinants, the little shapes that antibodies can bind to. Antibodies are characteristically like this Y shape, Y shape, Y shape. And it's cool that there's more antigenic determinants usually on an antigen because that means that we can have lots of antibodies binding. Lots of antibodies binding means that we get big results. Generally, B cells and T cells are made in mature in like 5-ish steps. First, they're made in the bone marrow. So that's the origin step. So there's an origin. They're made in the red marrow by that process, hematopoiesis in the red marrow. But once they're ready, the B cells, they kind of hang out, they mature in that red marrow while the T cells leave immature, swim through the blood into the thymus, and that's where they finish up their thing. In step 2, that maturation process is where we gain two qualities. I'm going to highlight them in pink. We gain the qualities of immunocompetence and self-tolerance. So to be immunocompetent, it's the presence of certain receptors that allow the lymphocyte to bind stuff on an antigen. Able to bind antigenic determinants. So if we're able to bind those shapes that we just looked at, then we would call that immunocompetent. The idea of self-tolerance is that whenever we see self things, we don't want the B cell or the T cell to bind our self proteins. So we want them to ignore those or be tolerant of the self proteins so they don't attack us. Don't attack us. The next step is that they seed the secondary lymphoid organs. So to seed those organs, it really just means we want the B cells and the T cells to be found everywhere. Before they're exposed to antigens, they don't have that much life experience as cells. So we usually call them naive. And once that's done, they'll encounter an antigen and then they'll get activated. Once activated, this signals the lymphocyte to divide and make lots and lots of clones. Clones are exact copies that will have bits on the surface of that cell that will recognize the antigen. And during this time of proliferation dividing and differentiation getting more mature, we turn into effector cells or the working cells that are actively getting rid of that antigen and memory cells. So we can turn into effector cells or we can turn into memory cells. Effector cells are the ones doing the thing, and the memory cells are the ones that hang out and remember so that there's a stronger and faster response next time. The T cells and memory cells, they hang out in the blood and the lymphoid organs, that kind of thing. While maturing, T cells and B cells, they must pass a series of tests in order to keep being those cells. This is important because T cells specifically need to recognize self proteins. So on the surface of our cells, so here's a cell, this is a self cell. We have self proteins. So whenever our body cells see this group of self proteins, then we're going to be like, oh, that's me. Sounds good. Those self proteins, they have a name. Those self proteins are called MHC. Those self proteins are called MHC. More on this later, but we want to make sure that the T cells want to recognize things that are presented on the MHC protein. So we have to pass two tests. We have to pass the positive selection test and the negative selection test. The goal of the positive selection test is that we want the T cells to recognize the self proteins. So here's our self proteins in purple. We want the T cell to be able to fully fit and recognize that self protein. So that's great. We want the T cell to make sure that we know it's us. The negative selection bit is that our self proteins have a little pocket where an antigen lives. We want to make sure that T cells don't recognize self antigens. So if the T cell recognizes that little antigen in that pocket of our self protein, that's no good because it's going to start attacking us. What we want is for that T cell to be like, what's going on in your little pocket, MHC complex? I don't understand. That's important because then the T cell is going to start to ignore us because the cells are ours. If this situation happened where the T cell recognized our self cells as bad, we would then have like an autoimmune disorder. If either one of these tests failed, the T cell could not continue being a T cell. I think this picture is really pretty, so I'm going to leave it on the screen while I talk about stuff and draw stuff. This is a scanning electron micrograph of a dendritic cell, which brings me to my next point about the different types of antigen-presenting cells. Antigen presenting cells, or APCs. This is a group of cells that engulf antigens and show lymphocytes pieces of it. They have surfaces on their cell that T cells can recognize, and that's really, really important, especially for T cells. There's three major types. There's dendritic cells. Those are the big guys. They live in the body tissues and they act like the bad guy delivery team. They catch antigens, phagocytize them a little bit, and then they show the T cells, and that usually happens like in a lymph node. There's also macrophages. Macrophages, they're another type of antigen presenting cells. This is because they present antigens to naive T cells, but they can almost just show T cells a little bit for fun in order to help keep T cells activated, to keep macrophages activated. And this is because lots of different stuff can go on, but we want to make sure the macrophages and the T cells are activated when they need to be. But also, this plays a role in promoting inflammatory responses when the macrophages show the antigens to the T cells. And then lastly, there are B cells, which we can consider a form of antigen presenting cell. They don't activate T cells directly, but they do present antigens to helper T cells. And that's done in order for the B cells to stay activated themselves, which we'll talk about. So how are B cells activated? So B cell activation. This happens when antigens, antibody generating substances, bind its receptors. But remember, adaptive immunity is a really specific thing. So this picture is showing you that this little red antigen is not binding to the B cell with the green on it. It's not binding to the B cell with the light blue on it. It's binding to the B cell with like the purpley dark blue on it. So this is a specific thing. So what happens is the antigen binds the receptor on a specific B cell. And so these other two guys, the green and the light blue, they stay inactive. And then they start to divide, they form clones, so exact copies of themselves. What happens is then we get we start moving on in the steps. Those clones then become either plasma cells or memory cells. So these guys are plasma cells. And remember that plasma cells make antibodies. And remember that antibodies have this characteristic Y shape. The memory cells, though, they just kind of hang out and remember. And we'll come back to that in just a minute. But the thing is that the antibodies, they'll hang out and they'll bind to that red antigen. And then once they bind to that red antigen, they mark it for destruction. The really cool thing is that if we see this antigen again, we won't have to start all the way up here. We'll actually start down here. We'll start with that memory B cell. We'll recognize the antigen and then start this process over so fast. And that idea that when we see an antigen again and our reaction is faster and easier, that's called establishing immunological memory. Immunological memory. And that's what this slide is showing you. We discussed in the last video how the adaptive immune system is really cool, except it takes a bit. But this is the idea of immunological memory. The blue line that we're looking at on this graph, or the greenish or whatever color that is showing you the antigen A, antibodies to antigen A. We just made antibodies for it. It took like a week from our first exposure to have enough antibodies, a little over a week. But by the second exposure to antigen A, look at how fast and how much that response is. So from exposure to maximum antibodies, that's a big difference. That secondary immune response is a lot bigger, and it's also sustained for longer. This is because the body is ready with those memory cells. Okay, more on humoral immunity. This is the antibody-mediated one. There's two major divisions here. There's active and passive. Active humoral immunity is when you see antigens and you make the antibodies against them. This is like a direct thing. It can be a natural thing, right? Like you get infected with something, you get sick, you get better, that kind of thing. Or you can encounter this artificially via something like a vaccine. Vaccines are usually like dead or attenuated versions of a pathogen. Attenuated means like weakened versions of a pathogen. The benefit here to the vaccine side of things is that you wouldn't have the getting sick part as you would with the naturally acquired immunity. The idea for both of these, though, is that you get through that primary response, and then you get to the secondary response even faster for next time. With vaccines, sometimes there's a booster shot that's given just to make sure that subsequent response is even bigger. The passive immunity side, though, is a little bit different because antibodies are introduced to the body rather than being homemade by like your own B cells. This is because the B cells don't make them, then you can't make the memory cells. You just have the antibodies, but they're not your own. It's kind of like borrowed antibodies. This can come naturally in fetuses, in infancy. While in utero, there's this organ, the placenta, which is the major nutrient delivery organ in the uterus, allows antibodies to cross over from the mother's blood into the fetuses. But it also occurs during infancy, during the consumption of breast milk, which contains antibodies from the mother. But again, these antibodies are mom's, they're not made by the baby. This can also happen artificially. This would be a treatment like things like antivenom, botulism treatment, rabies shots, tetanus shots. All of these are artificial antibodies to get rid of whatever that is, but the antibodies aren't yours. It's a short lived thing, too. It only lasts a couple of weeks. Okay, still talking about humoral or antibody mediated immunity. Antibodies, they kind of exist in this characteristic Y shape. So I think of antibodies as looking like this. We have like this big Y shape like this. Ooh. And that major Y shape is called the heavy chain. Each of those are called a heavy chain. And then there's like a little side piece to that Y, and that's called the light chain. That's called the light chain too. Okay, so this drawing is the same thing as this, is the same thing as this. So we have the heavy chain and the light chain. And within the heavy chain and the light chain, there's constant regions and there's variable regions. The variable regions are the ones that change. So that's how antibodies can be so individualized. They have a very specific structure, and these small changes can make them really different, like they bind to a different antigen. This is the cartoony version of an antibody, and this is like the actual protein structure. I don't really care that you know about the protein structure, except that it's important to remind you that proteins have a really specific structure, so any small changes can make them super different. All right, speaking of different antibodies, antibodies are this like Y shape with two chains, but because there can be different types or shapes of antibody, each can do different things. Antibodies are named like IG, whatever, and it stands for immunoglobulin. The IG, whatever, is immunoglobulin. That's one of those plasma proteins we were talking about in the blood unit. So remember, there's still going to be the heavy chain and the light chain with that little variation bit on the end. Oh, I should have mentioned on the previous slide that on the end of this Y shape, that's where the antigen binds. Okay, so back to this. So there's a bunch of different types of antibodies. The first are IgMs, which have the like big pentagon-like shape. They're really big. They're found in the plasma. of the blood, and they're really good at activating the complement proteins. Next, this image from your text is talking about IgAs, which is kind of like two of those Y shapes together. So it's called a dimer, which means like two things together. And these guys help pathogens not bind to things on the epithelium like mucous membranes. Then there's IgDs, again, immunoglobulin D. These guys are the ones that are found on the surface of the B cells. These are the ones that are like the B cell receptors. And this guy is where that antigen would bind. IgGs are the most common type of antibodies that we have. They're most abundant, like 75 to 85% of our antibodies are IgGs. And they're tiny. They can activate the immune system really rapidly and the complement system too. These are the guys that cross the placental barrier for that passive immunity we were talking about. And they're really especially active in the secondary immune response. The last one are IgEs. And the IgEs are really good at working with basophils and mast cells. When antigens bind to it, that helps those cells release histamine. So IgEs are really active in the allergic response. They don't hang out much though, they're not so common unless there's like a parasite or allergic response happening. Here's a nice little summary of all the things that humoral immunity does, and this is a great time to remind you about how antibodies work. Antibodies bind the antigen, and they can cause something like agglutination, like clumping up on that cell surface. And we saw these reactions when we did blood typing. We can also neutralize using antibodies, which means antibodies can block the sites where a bacterium or a virus would bind. And then there's also precipitation that can be done by antibodies. That makes it so whatever that substance is isn't dissolved in solution, it's taken out of that solution. And all of these actions lead to the same common goal, which is phagocytosis of that thing. So, in short, antibodies flag whatever it is for destruction so that phagocytes can eat it. Antibodies also activate the complement system, a group of proteins that turn up the volume on inflammatory response. But the complement can also lead to cell lysis by promoting water to go into pathogenic cells. The really cool thing about antibodies is that they have really specific targets they work on, which makes them really useful research tools. Using commercially available antibodies is super useful for blood typing, like we saw, but also this is how we diagnose pregnancies, hepatitis, rabies. There's lots of clinical uses, especially for things like cancers and to target really specific tissues. It's such a cool thing what modern medicine can do.